Okay guys, on this G-Block session we're going to cover three main ideas from year 12. It's going to involve a little bit of maths. So we're going to do cardiac output, we're going to cover mitotic index, and we'll cover index of diversity. So if we start with the definition of cardiac output then, it's all to do with the cardiac cycle, and the cardiac cycle is made of atrial systole, ventricular systole and diastole. So we've got three parts of the cardiac cycle, and that is a single heartbeat. So the cardiac output is really the volume of blood that is expelled or forced out of the left ventricle into the aorta, okay, per minute, so per unit time. So the equation is the cardiac output is equal to the stroke volume, and that is the volume of blood that's pumped out the left ventricle per heartbeat, times the heart rate, and that's how many beats we have per minute. So if we look at the stroke volume then, it's the volume of blood expelled from the left ventricle on one cardiac cycle from ventricular systole. That could be given to us in centimetre cubed, or it might be in decimetre cubed. The heart rate is the number of cardiac cycles or heartbeats per minute, and that might be given in seconds, so we might have to convert that to minutes. And the units for the cardiac output are normally given as decimetre cubed, which is a volume, per minute, so that's per unit time. So anytime we've got an equation made up of three parts and we've got two values within that equation, we can always work out the third value. So for example, if we wanted to work out cardiac output, we would need the stroke volume, and we'd also need the number of heartbeats per minute. So we can just use this triangle to help us there. Now, if we were given the cardiac output and the heartbeats per minute, we could work out the stroke volume. So the stroke volume would be the cardiac output divided by the number of heartbeats per minute. And if we wanted to work out the number of heartbeats per minute, we would need the cardiac output, and we would divide that by the stroke volume. Okay, so there's always a way of working out either the cardiac output or the stroke volume or the heartbeats, having been given the other two values. So if you guys want to pause the video at this stage and you can have a go at some of these questions, and then obviously when you finish, if you just click play. So I'll go through the question at the top, okay, in the yellow box, and it tells us the stroke volume is 70 centimetre cubed and the heart rate is 70 beats per minute. So we know the cardiac output is 70 centimetre cubed times 70 beats per minute, and that will give us a value of 4,900 centimetre cubed per minute. But we normally give the value in decimetre cubed. So if you guys just remind yourself that there's 1,000 centimetre cubed, okay, and that's equal to 1 decimetre cubed. So if you want to convert centimetre cubed into decimetre cubed, you just divide by 1,000. So 4.9 decimetre cubed per minute is the cardiac output. Now question 1 says 21 heartbeats in 20 seconds. So we can actually work out the heartbeats per minute. So we timed by 3. And that will give us heartbeats per minute. The volume of blood pumped out the left ventricle during ventricular systole had a stroke volume of 62 centimetre cubed. So we're given centimetre cubed as our volume, as our units. Now please be aware, whenever you do an equation, you should have the same units within the equation, and then you can convert the units at the end. So this is asking for the cardiac output in decimetre cubed per minute. So we can actually work it out in centimetre cubed and then convert that to decimetre cubed. So the answer to this one then is 21 heartbeats times 3 is 63 per minute. Cardiac output is 62 centimetre cubed times 63 beats per minute, and that will give us 3,906 centimetre cubed per minute. Divide by 1,000, and that will give us decimetre cubed per minute as 3.91. So we've divided by 1,000 to go from centimetre cubed to decimetre cubed. Now the second question then says if the cardiac output is 4.65 decimetre cubed per minute, and there were 41 heartbeats in 30 seconds, what would the stroke volume be in centimetre cubed? So because it's asking for centimetre cubed, we might want to convert decimetre cubed to centimetre cubed. So the cardiac output, we would times by 1,000 to go from decimetre cubed to centimetre cubed, and that's obviously per minute. And there's 82 heartbeats in 1 minute, because there's 41 in 30 seconds. So cardiac output is stroke volume times heartbeats per minute. Okay, stroke volume, if we rearrange the equation, is cardiac output divided by heartbeats per minute, and 4.65 times 1,000 is 4,650 centimetre cubed, okay, per minute, divided by 82 beats gives us 56.7 centimetre cubed per minute. So the key really is looking at the units. Does it want it in decimetre cubed or centimetre cubed? If it's asking for decimetre cubed and you've got an answer in centimetre cubed, you divide by 1,000. If you've got your answer in decimetre cubed but you want centimetre cubed, you times by 1,000. Okay, so now we're going to have a look at some data presented on a graph, and on the x-axis along the bottom we've got the venous blood pressure in kilopascals. So that is really the blood pressure inside the venules on the veins as the veins deliver the blood back to the right atrium. And on the y-axis we've got the stroke volume in centimetre cubed. So that is the blood being forced out the left ventricle during ventricular systole. Now underneath it says describe how the venous blood pressure affects the stroke volume. So with a describe question, it's really just using the data off the graph. Now you do not have to explain the data. You don't have to talk about the reasons why or behind why the stroke volume changes. Okay, it's just asking us to describe the data. So you don't just want to say the stroke volume goes up and then it plateaus. You've got to use the values a bit more carefully. So we can see when the venous blood pressure is 0 kilopascals, there's a stroke volume of 30 centimetre cubed on the y-axis. Now actually there's a steady rate increase when the venous blood pressure is increased from 0 to about 3 kilopascals. So hopefully you guys can see that the steady rate increase in stroke volume goes up until around about 3 kilopascals, and here the stroke volume is now 82 centimetre cubed. Okay, so this is what we call a steady rate increase, and it's from 0 to 3 kilopascals, and the stroke volume goes from 30 up to about 82 centimetre cubed. However, above 3 kilopascals on the x-axis, what you'll find is that the stroke volume remains at 82 centimetre cubed, even when the venous blood pressure is increased. So we know the stroke volume plateaus or remains constant above 3 kilopascals onwards or above 82 centimetre cubed. And the highest measurement we've got for the venous blood pressure is 8 kilopascals. And you can see that stroke volume at the top there. Look. So the mark scheme, if you guys just want to quick read through it, it says an increase in stroke volume at a constant rate or a steady rate as the blood pressure increases from 0 to 3 kilopascals. And that's up to 82 centimetre cubed, but it remains constant at 82 centimetre cubed, okay, from above 3 kilopascals all the way up to 8 kilopascals. Now the next question is going to ask us to use the x-axis, and it gives us a value of 2 kilopascals as the venous blood pressure. So what we can do is draw a straight line up where it hits the curve, then we go to the left, and we're going to take a measurement of the stroke volume on the y-axis. So at 2 kilopascals, that's going to give us a readout of 70 centimetre cubed as our stroke volume, and that's for the next question. So we can see that we're given the 2 kilopascals of the venous blood pressure, and we already know that we've read off the y-axis, and that's given us a stroke volume of 70 centimetre cubed. Now the question gives us the cardiac output, and that's 5,600 centimetre cubed per minute. And then it says calculate the number of times the ventricle contracts in 1 minute. So ventricular systole is a ventricle contraction, so 1 ventricular systole per heartbeat or per cardiac cycle. Okay, and how many times the ventricle contracts in a minute. So if we look at the answer then, we know the stroke volume because we got that from the table, from the graph. 5,600 centimetre cubed is the cardiac output per minute divided by 70 centimetre cubed. Okay, so the cardiac output divided by the stroke volume gives us 80 beats per minute, and obviously that's going to be our answer for 2 marks. Now the question underneath says explain how the blood in a vein in the leg is returned back to the heart. So we know the actual blood gets returned to the right atrium from the leg via the inferior vena cava. Now we know they're the biggest veins in the body with the biggest lumen diameter. So actually the blood pressure is very low in these veins that deliver the deoxygenated blood back into the right atrium. And if you think about it, obviously the blood's travelled the furthest distance because the blood would have left the left ventricle via the aorta, and it's gone all the way through the systemic circulation to get back to the heart. So you guys can pause the video and have a quick go at this question underneath if you wish, and then I'll just go through the answers. Okay, so the mark schemes then. Skeletal muscles that surround veins in the leg will contract. As they do so, they squeeze the vein, and that forces the blood upwards against gravity. But remember, we want to prevent backflow of blood down. So there's valves inside these veins in the leg. The valves open to allow the blood to go upwards through the valve, and then they snap shut to prevent backflow. So that ensures a single direction of blood flow back up to the right atrium. We know systole or systole of the ventricle pumps the blood out of the aorta, but we know actually the vein has the blood that's travelled the furthest distance. Okay, so actually when the right atrium is in diastole, when it's relaxed, after contraction, after systole, that's going to actually draw blood back up from the inferior vena cava back into the right atrium once it's relaxed. And we know the wide lumen of the vein provides little resistance to the friction of blood as it flows back up into the right atrium. Okay, so the next bit of maths we're going to talk about is the mitotic index. Now you guys have done this in the root tip squash experiment that you did in year 12. So you would have taken the tip of an onion root and you would have put it on a microscope slide. You would have crushed it under a cover slip very gently to get a single layer of cells. Now this is a light microscope image, and it's really important that the sample is 1 cell thick, so that allows the light to penetrate through the sample and it goes up into the objective lens. Now if you guys remember that the wavelength of light is actually a longer wavelength compared to the wavelength of electrons, so therefore the light microscope produces a lower resolution image compared to that of an electron microscope. Now with the mitotic index, what we can do is we can actually work out the length or how long it takes in minutes or seconds for mitosis to complete. And the way we do that is we count the number of whole cells in the image that are in any stage of mitosis. So it could be prophase, metaphase, anaphase or telophase. Now you know a cell is in mitosis if the chromosomes are visible because the chromatin is supercoiled. And we know that the chromatids that we can see are shorter and fatter, so they're visible. So we're going to count the number of cells that we can see with visible chromosomes, and we're going to divide that by the total number of whole cells that we can count in the image. And then you're going to times that by the length of 1 cell cycle. So please do be aware that the cell cycle is made up of interphase, which is made of G1, S phase, where you get DNA replication by semi-conservative replication, and then G2, where we get some organelles that replicate and increase ATP production. After interphase, the cell enters mitosis, which, like we say, is prophase, metaphase, anaphase, and telophase. Now, the other point that you might want to notice about most electron micrograph images or light microscope images, like we've got here on the left, is the scale bar. Okay, so you always want to look out for that scale bar, and that might allow you to work out a magnification. So if the actual length of the scale bar is given, you can measure the scale bar with a ruler, and that will give you the image size. And then if you want magnification, it's just image divided by the actual size. But please bear in mind you've got to use the same units for image size as you would do for the actual size. Okay, so this is the equation we're going to use. It's the number of whole cells that we can count in mitosis with supercoiled visible chromosomes divided by the total number of whole cells in the image times the length of a single cell cycle. So that's the complete cell cycle, and that's normally in minutes. Now, please bear in mind that it's not just a mitotic index for the length of mitosis. We can actually calculate the approximate length of each individual phase of mitosis. So if you counted the number of cells in prophase, for example, and divided that by the total number of cells in the image and timesed it by the length of one complete cell cycle, we'd have the length of prophase. You could find out the approximate length of metaphase, anaphase, or telophase by counting the number of cells in that stage. You divide that by the total number of cells and times by the length of one cell cycle. Now, what you'll probably find is that anaphase is actually the quickest of the phases in mitosis. So you'd expect fewer cells to be in anaphase compared to, for example, prophase, metaphase, or telophase. And because interphase is actually the longest stage of the cell cycle, you'd probably expect to have the highest number of cells, okay, as a proportion in interphase within your image. So you guys might just want to go back, remind yourselves of what interphase is. So it's G1, S phase for DNA replication or DNA synthesis in G2, and then you've got PMAT or PMAT, or the 4 stages of mitosis, all with, as we just said, visible chromosomes. So if you guys want to pause the video and have a quick go at these questions then. So it says a student was observing cells under a microscope. The student counted the number of cells she observed in each stage of the cell cycle. Of the 200 whole cells that she counted, only 6 were in mitosis. One cell cycle of onion root tissue takes 16 hours. Calculate how many minutes these cells spend in mitosis. So we're given hours, so obviously we want to convert hours into minutes. So 16 times 60. And then we can do our calculation. So 6 divided by 200 cells times the length of one cell cycle in minutes. So it's 16 hours times 60 minutes. And that's going to give us 28.8 minutes. And that's obviously our answer for 2 marks. Please always show you're working out, because you will get credit for that. Okay, in this question, it asks us to explain how you would calculate the magnification of a photograph. So obviously we're going to take the length of the scale bar in millimeters, that's the image length, and we're going to divide the image length by the actual length of the scale bar, which it tells us is 0.02 millimeters. Now obviously we don't need to convert units here because the length of the scale bar is measured in millimeters and the actual length is also given in millimeters. Sometimes obviously we are going to convert the units from millimeters into micrometers, especially if the actual length is given in micrometers, but that's not the case here. Now the question underneath then says the total number of cells at each stage of the cell cycle is counted. So we've actually got interphase, which we know is G1, S and G2, and that precedes mitosis. So actually mitosis is underneath here, and that's PMAT. And it says one cell cycle takes 24 hours. And it says, please do calculate the length of metaphase. But if you look at the answers, it's given us the units at the bottom. And it's not minutes this time, it's hours. So we don't actually need to convert the hours to minutes here. So it's actually even a bit simpler. So we know there's 12 cells that we've counted in metaphase. And the total number of cells is 200. So it's 12 divided by 200 times 24 is 1.44 hours. Now, please just be aware that you might have to identify the cells in metaphase, and you do that because each chromosome that you can see is a replicated chromosome, which means it's actually made from 2 identical sister chromatids joined at the centromere. Now, each replicated chromosome is attached to a separate spindle fibre via the centromere, and they'll be aligned down the equator of the cell. So we'll have, if it's a human cell, we'd have 46 replicated chromosomes. And obviously, if it's a plant cell and if it had 30 chromosomes, you'd have each of the 30 chromosomes on a separate spindle fibre aligned down the equator of the cell. Okay, the next bit of math is going to be about biodiversity. Now, we know we've got an index of diversity that we're going to have to calculate. And the reason that we're going to use index of diversity is because if we only had the species richness, that would be the total number of species present in a given area at a given time. So species richness does not involve the numbers of individuals. Now, if we had a look at these 2 areas, X and Y, even if we had exactly the same number of individuals of all the different species present, the data looks very, very similar. even though these are two different areas, two different habitats. So the benefit of being able to calculate the index of diversity is it gives us a little bit more information about the distribution of those species and the relative abundance of each of the species. So both habitats have the same species richness of 5, and we can see the total number of individuals is also the same. But if we actually look at the individual numbers of each of the 5 different species, what we'll see is a slightly different distribution. So we can see there's about even numbers of each of the 5 different species in habitat X. So there's not much variation between the different species here. But if we look at habitat Y, what we're going to see is even though we've got the same number of total individuals, there's a greater abundance of species D, because obviously we can see 36 is quite a lot higher than all the other numbers of all the other 4 different species. So it might be, for example, that species D is better adapted to habitat Y, and therefore it can outcompete members of the other species. That's interspecific competition for a given named resource. So we know that having the index of diversity provides us with a little bit more information about the distribution and the relative proportions of the individual species present in that community. And again, if we count the number of individuals, we can see that there is a difference here, and species D is most abundant in habitat Y. So one common question is to compare and contrast species richness with the index of diversity. So with compare questions, what we need to do is start the sentence with both have. And if it's the contrast part of the question, it will be what is the difference between? So we use whereas in the middle of the sentence. So if we're going to do the comparison then between species richness and index of diversity. So if we have a look at the answers then of the compare and contrast question, comparison now, we know both species richness and the index of diversity will actually take into account the number of different species given in that community. So for both measures of biodiversity, we need the number of different species present. And we know the contrasting statement then is the index of diversity takes into account the number of individuals for each of the different species, okay, as well as the number of different species, whereas species richness does not take into account the individual numbers from the different species that are present. So both have the number of different species, but index of diversity only takes into account the numbers of individuals for each of the different species, whereas species richness does not. Okay, so this is the equation that you guys have to learn and be able to use during an application question. And this is the index of diversity. So the D value on the left is the actual index of diversity. That's going to be a decimal normally. So it can be anywhere from, for example, 0.2 up to 2.8. So it's a value that you're given. The higher the value, the higher the biodiversity or the index of diversity for that given area. The capital N is the total number of organisms for all of the different species in that community, in that area. So if you have 5 different species and you count up the individual numbers in all 5, that will be the total number of organisms. The lowercase n is the total number of organisms for each of the individual species. So you have to do a number of different calculations on the bottom of the fraction. So n times n minus 1 you'll do for species A, then B, then C, then D, etc. So you can have a number of different calculations on the bottom, and then the sigma symbol, which is this one here, just means add the values together. So sigma means the sum of. So let's just use this equation again. We've got the sum of, we've got n times n minus 1 on the bottom of the equation, and we must do the calculation for each of the different species that were given in that community, in that area. So if you had 3 different species in a given community, in a given habitat, we'd have this expansion. So n1 is the number of individuals in the first species times n1 minus 1. So it might be 60 times 59. You do that for the second species. So there might be 120 individuals of species number 2. So that would be 120 times 119. And then you do the same for species 3. So it might be 78 is the total number of individuals of species 3 times 77. That's one less than 78. And if you had more species in that community, then obviously you'd have more calculations to do on the bottom of the equation. So what we'll do is use this index of diversity equation, and we'll use some data. So here we're going to have 5 different species, A to E. We're going to count the numbers of individuals of each of those different species. So just bear in mind, it depends on the type of species. You have done a topic on investigating populations, so please do bear that in mind. So if it's plant species, we could use random sampling with quadrats at random coordinates generated using a random number generator, where you place the center of each quadrat at that random coordinate. Now you need a large sample size to calculate a mean value. We could have, if it was animals, it might be mark, release and recapture, where you have capture 1, you mark them with a humane tag that doesn't harm the animal, or it doesn't affect their ability to reproduce, and it doesn't affect their feeding habits, or it doesn't make them more visible to predators. You then capture in the second capture, and you count the number that are marked within that. So that's mark, release and recapture. So once you've got the numbers of individuals of each species, we go n minus 1. So we can put those in, and then we go n times n minus 1. So 9 times 8 is 72, 10 times 9 is 90, 11 times 10 is 110. And then we add up those values at the end. So if you add up n times n minus 1 for each of the different species, okay, you're going to get, in this case, 454. We can then work out capital N times capital N minus 1 to give us the value that goes on the top of the equation. So the total number of individuals from our data was 50. N minus 1 is 49. And if you go 50 times 49, you're going to get 2,450. And that goes on the top of the equation. So we can then actually input the values. So we've got that on the top. And then we had 454 at the bottom when we actually added up the individual calculations for each of the different species. And that will give us an index of diversity of 5.4. Now, if we had a different area that we wanted to do index of diversity, we can do exactly the same process for the second area. So we could actually work out the index of diversity for 2 different areas or 2 different communities of organisms. So I'll let you guys pause the video, and you can actually have a go at this and just calculate it for habitat X versus habitat Y. This time we've got 6 different species A to F instead of 5, and you're given the individual numbers of each of those species in the 2 different habitats. So just pause the video and then you can have a quick go at the maths. Okay, so we're first going to calculate index of diversity for habitat X, and then we'll do the same for habitat Y. We then can compare the biodiversity by comparing the D values for the 2 different areas. So this is what we do for the first set of data. We've got species A to F, our 6 different species. They're the numbers that we had in the table. We go n minus 1, and then n times n minus 1. We're going to add up all of the values in the last column. That's going to be the bottom value of our fraction. The total number of individuals is 136. So you get that by adding all these values together, look. So that's 136. Capital N minus 1 is 135, times the 2 together is 18,300. 360, and that's going to go on the top of our fraction. So it's 18,360 divided by 3,464, and that will give us a value of 5.3. We're then going to do exactly the same for the second community. Okay, so species A to F. This time we're going to go n times n minus 1 for the 6 different species, and we get 2,342. We add up the total number of individuals of each of the 6 different species, and that's going to be 90. So 90 times 89 will give us 8,010. That goes on the top of the equation. 2,342 goes on the bottom of the equation, and that will give us a value of 3.42. Now we know 3.42 is obviously lower than 5.6, so it means that the second habitat, where we've got these 6 different species, has a lower index of diversity compared to the previous one. Now if we look at the data, we can see actually that the numbers found in the habitat, species D seems to be the most abundant of the species with the highest numbers of individuals. So it might be that species D can outcompete these other species, which is why there's higher numbers here. And it depends on obviously the type of organism that you're given in the question.